And I mean, what do you make? I mean, there is this idea that the human cortex would be so much different from non-mammalian cortices. So how do you approach them? Are you looking into that, or how do you think working on salamanders would be predictive of such a different structure?
Yeah, it's true. There are obvious differences. A large part of the mammalian cortex is called neo-cortex because it's new in evolution. But however, many of these definitions have been based mostly on neuroanatomical comparisons, counting how many layers are in there. And what we've been able to do with using comparative single-cell RNA sequencing is to really look at the cell type level, defining cell types from their gene expression profile, and being able to compare cell types across species. Mm-hmm. And I would say from the work that I've done in Frankfurt and the work that now my lab has continued, there are two lessons. And one is that there are certain types of neurons and brain areas that are deeply conserved or extremely similar, going at least in the salamander, in reptiles and in mammals. But the neocortex of mammals is indeed composed of glutamatergic neurons that have gene expression profiles that are unique to mammals, entirely new. So the most ancient parts relate to things like hippocampus, which is involved in memory and spatial navigation, or to the piriform cortex, which is another part of the brain that is involved in olfaction. But once you look at the neuron types that make up the 6 layers of the neocortex, you can see that indeed there has been a lot of cell type innovation in that part of the brain.